This is Trimontia Genomics' prototype of the Cheetah Genome Browser, demonstrating its capabilities of displaying high-throughput sequencing data. The advance of high-throughput sequencing technology allows scientists and clinicians to read entire genomes, including individual genomes of patients, rapidly expanding our knowledge of sequence variations and mutations associated with disease. Sequencing technology is also increasingly employed in powerful new methods that not only identify sequence variations, but also reveal how all sorts of genetic, physiological, or environmental factors affect gene activity on a whole genome level. These technologies have created an explosion in sequencing data that researchers are struggling to characterize. New tools are needed to deal with this unprecedented volume of information. Here, we present real experimental data that examine how cells of the immune system react on encountering bacteria to induce inflammation. This type of data is aligned to the genomic sequence and then is represented graphically down to a single nucleotide resolution. These graphs show in orange, green, and blue indicate gene activity at 3 different levels. Each of these is independently measured at 12 different time points, represented by increasingly darker shades during the first 2 hours after bacterial exposure. Even at this prototype stage, the Cheetah Genome Browser can be used to perform tasks virtually impossible with currently available tools. For example, let's find genes that are activated upon encountering bacteria. We will superimpose the corresponding graphs with time point 0 at the front and time point 120 minutes at the back. Since time was represented in increasingly darker shades, the genes that are being activated now appear darker than the rest, and we can easily spot several of these here. Let's now examine one of these in detail. As we zoom in, we find 3 genes in this region. The one on the right does not seem to be activated, unlike the other 2 that appear darker. Focusing on one of the activated genes, we can now explore the kinetics of its activation in detail. Normally, we would separate the superimposed graphs to examine gene activity. In this case, we would notice that the activity of this gene increases over time at all 3 levels examined here. With the Cheetah Genome Browser, we have a novel way to examine this time course, which allows us to keep the graphs condensed. Since the Cheetah Genome Browser renders all data 3-dimensionally, we can simply rotate the image and take a look at them from another angle. In this view, the horizontal axis represents the time that we can easily see that this gene becomes highly upregulated during the early response to inflammation and then declines by the end of the 2nd hour after the stimulus. With the fast-paced graphics engine, we can very easily repeat this task. Let's zoom out to a chromosome view and find another activated gene. Looks like this gene follows a different kinetics compared to the previous one. We can use the 3D view to confirm this. Indeed, the activity of this gene increases throughout the 2-hour period, reaching a maximum at the 120th minute. Now let's test other interactive features of the browser using another experimental dataset. Here we compare gene activity in 2 populations of neurons, normal cells and neurons lacking an important regulator linked to autism and epilepsy, called RB-FOX. We zoomed into a gene important for transmitting nerve signals, which is active and fully functional, but not in developing neurons, as can be seen here. This shows us multiple sites of RB-FOX interaction within this gene, located close to the center peaks. There is another binding site at the end of the gene for a different RB-FOX isoform. Zooming in on the central part of the gene, we notice clusters of RB-FOX binding sites on each side of the central part of the gene. We can now see that the overall gene activity is not significantly altered in neurons lacking RB-FOX. Instead, only the height of the central peak is greatly reduced compared to control neurons. Importantly, this product is known to alter neuronal transmission in the brain. Thus, using the Cheetah Genome Browser, we can rapidly identify RB-FOX regulatory targets, which contribute to neurological dysfunction upon loss of this regulator. We can now try some useful 3D features of the Cheetah Genome Browser. Switching between the opposite strands of the genomic DNA is now achieved by simply flipping the view at 180 degrees. We can also change the order of the superimposed tracks using simple intuitive commands. Trimontia Genomics' ultra-fast graphics engine allows these functions to perform equally well on a single gene or at a full chromosome view. In conclusion, we are excited to share with you our vision for the future of genome data visualization and our passion to create a tool that will be instrumental in the rapidly expanding field of genome research.